Thank you, everyone, for joining us today for the very first EMSL Learn webinar series presentation, where we're going to be discussing supercomputing your data. Just a couple reminders. In the link, we're going to drop a link to check out the recording of this today's webinar on our EMSL Learn portion of the website. If you want to go back and hear something, or if you want to send it out to your friends, you can do so through that web link. We will have it up hopefully by the end of today. Second, next month, we're going to be talking about visualizing root architecture. So if you are interested in joining us for that webinar, the link to registration for that one is also in the chat. And then also to keep up with the latest news at EMSL, subscribe to our channels. You can get emails sent to you directly about events, these webinars, our science highlights, calls for proposals. Also check out our social media channels. All three of them are also going to be posted in the chat window: Twitter, LinkedIn, Facebook. So without further ado, speakers for today are Kurt Maier, who's going to be talking to you about supercomputing resources at EMSL. Amy Jystad, who will be talking about how her group used EMSL's supercomputing resources for identifying small molecules in complex samples. And then Jay Bardhan, who will be talking about accessing or how to access our supercomputing resources, namely TAhoma. And he'll be discussing a recent success story. So I give the floor now to Kurt Maier. Thanks.
Thanks very much. So I'll be giving a little bit of an overview of what supercomputing and related resources. Sorry, I guess my video wasn't starting on time. So I'll be giving a little bit of an overview about what supercomputing and related resources EMSL has available for users, and then talking a little bit about our primary HPC cluster and the technical information that went into its design and what it looks like. So we can go to the next slide. So the centerpiece, of course, is our high performance computing resource, which is TAhoma, and that is heterogeneous supercomputing. What that means is we've got more than one kind of processor and node architecture built into the same cluster. Associated with that, we've got data management resources, and that includes Aurora, which is our quite large multi-petabyte data archive, which is tape and disk based, and is available for use for any data generated as part of a given project. And data management also covers our Nexus tool, which is sort of, those of you who've worked with EMSL before know that we have a project lifecycle management tool, and it's got an inbuilt archive data organization system in it. So those of you who've heard terms like EUS or MyEMSL, those are now built into the larger Nexus project. But it's not all HPC and data management. We also have various other resources in order to support different kinds of applications and user goals. And so we've been expanding our cloud computing offerings recently. So we still offer classic VMs for serving specific kinds of application data. But now we've also got a Kubernetes cluster built out on site, which is locally managed, and that enables us to deploy containers and more complicated orchestration platforms more readily and ideally connected to the data on site. So we can go to the next slide, and I'll talk a little bit about TAhoma. TAhoma is 160 compute nodes and 24 analysis nodes as far as the workhorse part of the cluster. And I'll get into more detail about what the differences are between those types of nodes in a little bit. Those nodes are all connected to each other with HDR InfiniBand. This is a networking technology, and it's one of the things that makes a cluster is super fast, low latency, interconnected network, so that any node can talk to any other node at a high rate of speed. And so the rated speed for this particular network, this fabric is what we refer to it as in the cluster, runs about 100 times faster than the wire that comes out of the wall and goes into your desktop. That's speaking from a throughput standpoint, but it's also much faster in a latency standpoint, in that your normal day-to-day networking latency, i.e. how fast data gets from one machine to another, tends to be measured in milliseconds, or if you're real close to the network, possibly microseconds. Whereas InfiniBand and similar clustering fabrics, that latency is measured in the nanoseconds, and usually tens of nanoseconds. So the other thing that makes a cluster is some shared storage. So we've got a 10 petabyte file system that is connected into the fabric, the same HDR InfiniBand, and it's mounted and available on all of the compute and analysis nodes, as well as the login nodes. And that's a high performance shared file system. So every node in the cluster has the same picture, the same viewpoint into that file system. And so it's a great place to where if you've got a couple dozen nodes working on one task, you can write data on one node and read it on another node in short order. We'll talk a little bit more about storage strategies and IO, but the fabric, the interconnect, and the shared storage are really the two factors that make a cluster. So every node, all 184 compute and analysis nodes, are connected with this high-speed fabric and have access to this shared file system. So we can go to the next slide. And so here's the technical details about those nodes. The majority of the nodes are compute nodes. They each have 2 processors. Each processor has 18 cores, and each of those cores runs at 3.1 gigahertz. Now, each node has 384 gigs of RAM and 2 terabytes of local storage. And this is different than the 10 petabytes of shared storage. This is storage that's built onto the node, and that's NVMe, local to each node. So the philosophy behind selecting this particular layout is that we noticed that not all jobs that we're running on behalf of our users, not all jobs scaled wide. In other words, not all jobs needed many nodes. Some of the jobs were what we call single threaded, meaning they needed a single core or maybe even a handful of cores on one node. But they benefited from a higher clock speed, meaning that they could do more instructions per second. The job would run faster per node with a higher clock speed. So we tried to strike a balance between having a lot of cores and so that programs that can make use of that parallelism have it available, and a high clock speed so that programs that didn't necessarily benefit from wide scaling could still get performance benefit from running on the cluster. And the reason that we've got that specific number of gigabytes of RAM is because we mandated in the design document that each memory channel, which are the electric pathways that the processors use to access the memory, was fully populated with chips, so that there's an even performance characteristic, no matter where your job is, or where on the physical hardware your program might be running and where in the physical memory hardware the data might be. We've got the highest possible throughput for memory IO. And so that wraps up the compute nodes. We also have 24 analysis nodes, which are the same base profile as the compute nodes. They've got the same 2 processors, same 18 cores, the same 3.1 gigahertz clock speed, but they also have 2 NVIDIA Tesla V100 GPUs. And they have much more RAM, same memory layout, but bigger DIMMs. So we've got 1.5 terabytes of RAM per node with much more local storage on board. So we've got 8 terabytes of per node storage in addition to the access to the clustered storage. So the idea here obviously is that if we've got bigger data sets that can't fit in 384 gigs of RAM, these nodes can generally hold it. And you've also got, you can also use that RAM for staging data to get it to and from those GPUs, because those are sitting behind a PCIe pipeline. So it's good to be able to stage data. Now, your CPUs and your GPUs work best on different kinds of tasks. The CPUs are obviously general purpose processors that Intel has designed to be as efficient as possible at all kinds of workloads. The GPUs really benefit from specific sorts of workloads. So vector math and matrix operations and that family of mathematic operation generally happen. You get the most benefit out of the GPUs from that kind of work. So in order to best select which kind of node your job runs on, it helps to have an understanding of the runtime characteristics of your workload, and that's something that we can help with. Obviously, we don't leave everybody out in the cold to decide what kind of hardware will magically make anything run faster. We've got expertise to advise you on that. So that's sort of the philosophy here, is that we've got high clock speed nodes that'll run low core count jobs efficiently and quickly, and then we've got big memory nodes with GPUs attached to them for specific kind of data sets that can benefit from NVIDIA acceleration. And obviously we're willing to work with people to figure stuff like that out. So if we go to the next slide. Yeah, that's the end of our slides. Great. So at this point, do we want to do questions now, Linda, or should we wait till the end of the presentation?
Let's wait till the end.
Okay. In that case, I'll hand things over to Amy, and I'll get out of the way.
Great. Thank you, Kurt. Hi, everyone. I'm Amy Jystad. I'm a postdoc here at PNNL, and today I'm going to be talking to you about some of the tools that my team, Hydra, has been creating for the identification of small molecules in complex samples. Next slide, please. So the typical protocol for identifying molecules in experimental measurements requires the use of reference libraries that you can compare your measurement against. However, you can imagine that this might run into some issues along the lines when we recognize that the number of known molecules that we actually have compared to the very vast molecular universe is really, really small. And an additional problem when we want to then build our reference libraries comes into play when we recognize that an even smaller fraction of the known molecules is available for purchase. And so you are generally just rather hindered when you want to build a reference library. And if you ever run into the case where you have a molecule in your sample that you don't necessarily have a reference for. And so a lot of the tools that we have been building within our Hydra team has been to try and remove this dependency on reference libraries. Next slide, please. And so we're hoping to do this in a two-pronged approach, combining both experiment and computations. You can see on the top portion of this figure where we would take machine learning, quantum chemistry, whatever quantum calculated capabilities that we may have to build a database of properties that we can then take to compare against properties we can elucidate from experiment, which would be then the bottom portion of this figure. And then bringing these calculated and experimental properties all together to match them up together right there in the middle circle. Next slide. And so today I'm going to be discussing 3 of the tools that we have, specifically because they are ones that we use EMSL HPC resources for. I'm going to be starting with Icicle, which highlights the section in the blue, and primarily focusing on Icicle today, but then briefly touching on DEMOS in the green and MAME in the red. Next slide. beginning with Icicle. Yeah. So what we would like to do is develop this library of properties that are calculated in parts with DFT and, for instance, IR, mass spec collision cross-section properties, NMR chemical shifts, and so on. And the actual process in order to go through this workflow is pretty standardized. You start with your original geometry, you get your conformers of those geometries, you optimize the structure with DFT, as well as maybe calculating the properties with DFT, and then following it up with any data post-processing we might need to do. And the issue that we have with this process here is that there's a number of different softwares that you actually need. So the first step, you might use RDKit and ChemAxon, then you use a molecular dynamics suite of AMBER, XTB, whatever, an electronic structure package then, and then whatever data manipulation you use, Python, Pandas, Excel, whatever floats your boat. But you would need to actually pass off the information between each of these steps. And if you want to build a library of hundreds or thousands of molecules, you can imagine that this hands-on pass off is going to be a very, very tedious process, which I can tell you from experience is. Next slide. And so we have addressed this issue by developing the in silico chemical library engine, or Icicle, where we've automated this process to remove a lot of the hands-on time required by the user. Next slide. And so for an example, really all the user would have to do is to compile a list of either INCHI or SMILE strings of molecules of interest that they have. Icicle would then take this information, convert that string into a two-dimensional, three-dimensional geometry, see if there's any sort of molecular manipulation we need to do, for instance, if we need to desalt it. And then once we have that geometry, Icicle will then just automatically take that structure through molecular dynamics for conformers, automatically take those conformer structures through DFT, then automatically process or calculate the properties that need be. And then once the DFT is done, we can parse out the necessary values. In the NMR case, we parse out our isotropic shielding values and then conduct our own Boltzmann weighting so that ultimately you end up with just one set of chemical shifts per molecule, not n sets. But overall, just removing a lot of the hands-on work that the user would generally need to do beforehand. Next slide. And so we've been able to apply this to two cases thus far. We've built a NMR library of about 300 molecules, and we approached it with 8 different levels of DFT, which, as you can imagine, is a very computationally expensive process, thus requiring the need for EMSL resources. And we've also done this process for collision cross-sections of 1,300 compounds as well. My primary work here at PNNL, not yet. My primary work here at PNNL is to take Icicle and apply it to natural products, as we're in the process of developing a database for natural products magnetic resonance measurements of from both calculations and experiment in this npmrd.org, or Natural Products Magnetic Resonance Database. And our ultimate goal with this is to be able to compile all of these calculated and measured values for about 350,000 molecules. And our hope, our dream one day, is to actually have a new user deposit a new structure and their chemical information or chemical shift information. And then we would run a DFT calculation to compare the calculated versus their experimental and to help determine how well they've identified that structure. Next slide. So now that we have our database of properties that we obtained from calculations, for instance, from our Icicle piece of software, what about from the experimental side? Next slide. And so with this, we've developed DEIMOS, or our Data Extraction Integrated Multidimensional Spectrometry tool, which takes in three-dimensional raw experimental data from a mass spec, which you can see is roughly plotted there on the left. And it compresses it down first into 2 dimensions, which you can see on the top right, and then once again into 1 dimension, as is on the right. And from this process, we're able to back out properties that we compare against our calculated, such as the mass over charge ratio, the drift or retention times as well. The thing is, because this initial piece of data is so large in terms of memory, for instance, just one component of this measurement can require up to about 200 gigabytes of memory. We do use EMSL computing resources to conduct this process. Next slide, please. So now that we have our calculated properties and we have our experimental properties, we're now going to bring them together to actually help identify the molecules in our sample. Next slide. So we've done this by creating our multi-attribute matching engine, which takes in output from our Icicle or SAPEX and SAPEX. Next slide. Takes in output from experimental samples that can be processed using demos. In our proof of concept study, we obtained 10 samples from the EPA, got their similar properties, and then next slide. Brought it all together to apply our multi-attribute matching engine, and so that we could identify the molecules within the sample and apply a score to identify just the probability of these molecules in our calculated library and identify how likely they are to be in our experimental sample. Yes, next slide. And so we were able to do this with these 10 mixed samples, 750 with required DFT. And overall, we were able to have a 91% prediction accuracy rate without having any need for an experimental library of reference molecules. Next slide. And so I've touched on 3 of the tools that we've used on EMSL HPC resources with the intention of this small molecule identification and mixtures without the use of standards. But we have developed a number of other tools, which I have listed on the left side. And we're more than happy to talk about and share any of these tools. And if you have any interest in applications or questions, please feel free to contact us. We do have a new team email as hydra@pnnl.gov. And so please feel free to contact us there. Next slide. And so to finish out, just like to acknowledge NIH for funding for all these projects, John Court and Ryan Renslow for mentorship and guidance, collaborators, and all of the lovely, lovely people that are working with me at Team Hydra. So thanks. And from here, I can pass it off to Jay.
Hi, everyone. Thanks very much, Amy. That was really cool. I am a computational scientist here at EMSL, and I am the lead scientist for one of our integrated research platforms focusing on computing. Today, I'm going to talk about 2 topics. One is how to access TAhoma, and the other is a recent success story that should make the case as to what kind of value we can bring to help you do more science in less time. EMSL is organized into 3 science areas shown here: environmental transformations and interactions, functional and systems biology, and where I sit, computing, analytics, and modeling. Next slide, please. These 3 science areas serve both sides of the BER research house, so to speak. And at the bottom, you can see 7 integrated research platforms. Each of these is focused in a particular area of scientific investigation relevant to BER, and systems modeling and data sciences, where I work, supports modeling, computing, and analytics in all 6 of the other IRPs. Next slide, please. All right, so accessing TAhoma, our new and fancy, very cool, very fast supercomputer. There are multiple kinds of user proposals that you can submit to EMSL. User proposals are the main mechanism to access TAhoma. The website is shown at the upper right, and on the left is a chart that taken straight from the website that illustrates the different kinds of user proposals that EMSL accepts. The ones that are grayed out do not offer access to, or the ones, sorry, with the Buster logo do not offer access to TAhoma, nor are they anticipated to in the future. The important thing to take away from this is that there are a variety of types of user proposals. They have different durations shown in the right column. There are different times of year when we accept proposals of those types. And some of the proposal mechanisms are only offered at specific times, and the topics may change from year to year. One important thing to note is that the mechanisms have varying degrees of competitiveness and offer different amounts of staff support and different amounts of computer time, to put it bluntly, on TAhoma. Next slide, please. Thank you. Our three large or main kinds of user proposals are the large-scale research, the ficus proposal mechanism, which is always associated with another user facility, and you need to have a research project that would need the unique resources of both EMSL and either JGI or ARM. And the third mechanism is the exploratory research call. So the basic process for submitting a user proposal and obtaining time on TAhoma is sketched out here. The main categories of steps are shown in the chevron, and then a little bit more detail below. The first step is that we open a proposal call. The topics for any given call are developed by EMSL scientific leadership, and we in consultation with BER program management. And once the program calls are written and released, they're advertised through digital media, social media, peer networks, and all that. And the first stage of pursuing that proposal call is to submit a letter of intent saying, here's roughly speaking the kind of research that I would like to do. Here's roughly what I think would be needed to do it. And here's why it is important and aligns with the topics in your call. We highly recommend that if you're interested in submitting a user proposal, that you first talk with the relevant IRP lead scientist. So for computing, that would be me. As well as we can connect you with other scientists, whether that's staff who are expert in that kind of modeling or computing, or instrument scientists if you are working on projects that have both experimental and computing components. Those consultations will help you align the scope of the research that you're talking about, as well as to make sure that the subject is well aligned with the call topics. After you submit the LOI, it's reviewed internally for responsiveness to the call topics and feasibility. Then a select number of those are asked for full proposals. We recommend further consultation in more detail for your precise resource needs as best you can estimate. Talk about different trade-offs of how would certain resources affect the scale of science or what you might need to do. Once your full proposal is submitted, there's a series of internal reviews for environmental safety and health, for internal administration, technical feasibility, and so forth, before they're sent to an external peer review panel. And that panel assesses and provides a ranking. And then the IRP lead scientists work with other folks within EMSL to estimate the resource needs, balance against available, one scientist only has so many hours in a day. And we also do cost estimates because we have a budget that we have to keep within. We then award proposals in ranked order within that support budget and make awards. And at that point, if you're awarded a user project, a project manager, and the relevant IRP lead scientists will reach out to you to coordinate a kickoff meeting with you and your team to highlight how to get started. Okay, next slide, please. So with all of that, I do want to highlight that one of the aspects of computing at EMSL is that we have a unique combination of resources and expert support that can cross a wide range of areas of computing sciences, modeling, subject matter expertise, and so forth. So I'd like to highlight that with a recent case study that's a success story that utilized TAhoma's hybrid architecture, and that is analyzing metatranscriptomics data from wetland soils. Next slide, please. The team that was doing this study was looking at microbial communities using metatranscriptomics. For those who aren't experts in metatranscriptomics, here's the rough story. You know that an organism's DNA is stored in its genome, and that the DNA is transcribed into RNA, which is then translated into protein. So if we wanted to study what an organism could do, we might study its genome. And if we want to get a better sense of what it's actually doing, we might study its transcriptome, what DNA sequences are being actively transcribed into RNA. That works well if we're talking about single organisms, consider the Human Genome Project. For complex samples, like in environmental studies, we may not be able to isolate one single organism that's of interest, and we may not even know which organisms are in our sample. In that case, we might look at the whole sample by blowing apart all the cells and sequencing either all the DNA, giving us a metagenome, or all the RNA, giving us a metatranscriptome. The goal of all of this experimental work of this particular project was to identify the activities of the different proteins in these samples. And in order to understand nutrient cycling or biogeochemistry of these soils. And the way that that's done is by assembling the small RNA sequences into larger pieces, as large as can reasonably be done, and then comparing those larger DNA sequences to genes where we might actually know both their sequence in full and an associated function or a predicted function. The challenge for computing is that the data sets in these samples are huge. It's 200, for this study, it was 270 million reads or small sequences of RNA. It's not one big puzzle with 270 million pieces. In a puzzle, you know what the picture is, generally speaking. You know that each piece belongs somewhere and that there aren't duplicates, and you're hopefully not missing too many pieces. Here, we don't know what the pictures are. We don't know how many puzzles we're trying to put together. Lots of pieces are going to be missing. Lots of pieces are going to be duplicates, and there are going to be pieces that just don't fit ever because they're noise in the experimental study or in our measurement system. So it's a really hard assembly problem. Next slide, please. As a result of all of those challenges, an earlier analysis that the team had done had taken months to produce. That's a lot of manual work. It's a lot of care, care that needs to be taken to get every step correct. After they worked with the TAhoma team, TAhoma was able to run a complete analysis of the same set of samples in 24 hours. So that's sort of two orders of magnitude, 100-fold acceleration in the science. And as we'll describe, that can make an enormous difference in the kind of approach you take to analyzing your data. Next slide, please. So here's how it was done, loosely speaking. The software that the team wanted to use to do this metatranscriptome analysis is called Trinity, and it has two distinct phases. One phase where you're sort of clustering similar or related reads, the sequences, needs a ton of memory, but it isn't very parallel. And the second phase, after you've done all this clustering of small reads, involves a lot of very small independent tasks, each of which doesn't need much memory, but you have millions of those tasks. So that let Dave Cowley, who was leading this work, make use of TAhoma's heterogeneous design. We had on the large memory, 1.5 terabytes of RAM analysis nodes, we could run that first phase. And for the second phase, well, they're independent tasks. So we could use dozens of TAhoma's regular compute nodes, where the 300 plus gigabytes of RAM was adequate. That got the time down from months to a couple of weeks. But Dave looked at the usage statistics and the logs and all that and said, you know what, we can squeeze a lot more performance out of this. So he reached out to the Trinity developers, the component that lets Trinity run on high performance computing hardware. And they worked together to identify a key optimization, which got the analysis time down from 2 weeks to under 24 hours. So the point to draw from this is that getting top performance requires some expert knowledge sometimes. And that's really, really important because in any given project, you may be allocated a certain number of hours to run on TAhoma. You want to get as much science as possible out of that set amount of hours. Next slide, please. So now I'd like to tell you about the impact that acceleration had on their science. One way that you can assess the quality of the assembly that you've done is by saying, all right, so I had all these 270 million pieces. How many of those wound up in a partially assembled puzzle? Now, when your assembly process takes months to produce, and you say, well, I was doing a study just up the river, right? It was only a mile up the river. It's probably a pretty similar environment and set of microbes. That might save you months of work. But when you do that, you find that only 4 to 7% of the reads align. They only 4 to 7% fit into that previous assembly. So if I have 25 puzzle pieces, only 1 or 2 on average was actually being used. But if I can do the analysis in 24 hours, it turns out that over 40% of those reads then align with that site-specific metatranscriptome. That's a huge difference in the amount of data that you're actually using from the field. Now, zooming out to the overall goal of the study, which was to predict biochemical activity of these microbial communities, the overall metatranscriptome allowed the prediction of over 1 million genes. And in turn, when you look at what genes we know what they do, or we think we know what they do, that yielded over 260,000 informative gene annotations. So in comparison to using the previously used metatranscriptome, where we had only 4 to 7 reads aligned, they could only obtain a roughly 100,000 annotations. So by using this faster high-performance cluster, they were able to more than double the amount of information and understanding that they obtained from the experimental work that they had already done. Next slide, please. So thinking more broadly about what this kind of capability now enables, the team wrote a summary of this work and said that the ability to assemble large-scale metatranscriptomes in a day at high accuracy can significantly advance EMSL's research of complex environmental communities by supporting the analysis of a large number of samples, that is, it only takes 1 day per, and iterations under multiple conditions, for instance, at different time points. So there are a wide number of applications for this. And what we really wanted to do here was highlight, as a complement to the molecular scale studies and simulations that Amy presented, here's a study of analyzing environmental scale data. I believe that that is the end of my section. Next slide, please. And we're happy to take questions. Thank you.
All right. So thank you, everyone. You sent us quite a lot of great questions. So we'll just start with I'm just going to go down the list. So Kurt, I believe this one is for you. Are the 2 GPUs linked by NVLink? Can the memory on them be pooled?
So the NVLink functionality is supported by the hardware, but we don't have the NV bridge installed, from what I can tell. So the P2P functionality, system memory, atomics and access, that stuff is supported, but the SLI functionality is unsupported. So as far as pooling memory, you'd have coherency between system memory and the 32 gigabytes of onboard memory per GPU card, but there's no SLI functionality between them.
All right. The next one's also for you. Is TAhoma unique among all DOE high-performance computers? Are many others like it out there? Which are most powerful?
Yes, TAhoma is unique. I'll put it that way. It's hard to speak on behalf of the entire DOE HPC catalog, but you know, the headline systems, Sierra, Summit, the new Pearl Mutter system that NERSC's putting in, these are much larger systems, and of course most of the clusters that run under the Oscar program have sort of a dual mission between supporting scientific computing and sort of pushing the boundaries of what HPC can do. And TAhoma was designed to be a workhorse. It was focused on the jobs that we saw running, the needs that our users have today, and really dialing in exactly what sort of design would benefit people directly with existing workloads. So it's sort of a different application of HPC than some of the bigger top 500 entrants out there. But it is unique because we designed it to meet the needs of EMSL users specifically.
All All right, next one, Kurt, again for you. How much VRAM does the GPU have, and how much, and does it matter much?
So each of those cards, there's 2 per analysis node. Each card has 32 gigs of VRAM. It's hard to say if it matters much. If you're compute constrained, then it might not. But if you're memory constrained, it might matter a lot. So it's workload dependent. But basically, it means that you can fit 32 gigs of working material on that card at once. And of course, reaching back, one of the advantages of the CUDA acceleration is that it's got atomic access to system memory. So getting data down the PCIe pipeline into that 32 gig space is relatively fast, and CUDA handles that sort of memory moving pretty well. So it really depends on how good your software tool set is at taking advantage of the architecture.
All right, next question for both Kurt and/or Jay. What types of sample analysis at EMSL require the highest computing speeds and power?
I'll say a sentence from my experience, and then Jay can chime in if he may know better. But the cryo-electron microscopy work produces huge imaging data sets that need a pretty good bit of horsepower to resolve into usable data sets. So in my experience, that's one of the as far as instrument analysis, that's one of the more heavy lifts we've got as far as compute power.
I would not argue with that.
All right, next one is for Amy. So Amy, how easily does Icicle perform these NMR calculations for different solvent or magnetic field conditions in this automated way?
For the magnetic field conditions, the easy answer is that we don't include that parameter in our calculation. For the different solvents point of view, you can actually tailor it to whatever solvent that your system is. We do have a default run or default parameters for running our system. I think the current default solvent is DMSO. But there are other files that the user can edit, and one of the things that they can edit is the solvent that they are interested in. So very easily.
All right. So I think this one is for Jay. How can I apply this for immuno-oncology?
It depends quite a bit on the precise nature of the project. Medical research is not what TAhoma is purposed to do. So the scope of the work would be something that I'd want to learn more about. Certainly large-scale molecular dynamic simulations, multi-scale simulations involving both quantum mechanics and MD are very much in scope. But things like antibody design sort of fall into a different category. We are supporting work that is related to COVID-19, but that is a little bit of a special case. So I need to learn more in order to be able to provide a really clear answer on that.
All right. Next one is for you again, Jay. On the BER atmospheric side of things, are there any TAhoma related projects underway that collaborate with DOE's Atmospheric Radiation Measurement, or ARM, user facility? There have been some collaborations historically related to atmospheric aerosols. So just wondering if there's any new projects using TAhoma at this moment.
There have been. I'm not off the top of my head able to recall any new projects that are starting there, but absolutely, if folks are interested, there is a FICUS ARM call every year, and we would welcome that.
All right, another one for you, Jay. what's the computational requirements you had to run these tools? And I think they might be referring to Trinity, for which you used an EMSL cluster.
I'm not sure I'm following the question. Could the person adds, that question is for Amy. I'm off the hook.
Oh, sorry. It was for Amy.
For the Icicle component, mainly just need a number of parallel nodes that are able to be run in parallel. Same with MAME and Deimos. It's really just a memory requirement, as far as I know. Jamie or Sean Colby, Jamie Nunez or Sean Colby would know better for MAME and Deimos.
All right. So Kurt or Jay, does EMSL support Cromwell WDL workflow management system? Does any project use WDL on an EMSL cluster?
There's been some talk about various workflow management systems. As far as WDL specific ones, I'm not positive off the top of my head that we've got support for any of those already installed. But if I remember correctly, Cromwell is one that was originally written for the Google Compute Engine. And so I'm pretty sure it's containerizable, and we've got the capability to run Singularity containers on the cluster. So usually the way a WDL runner would operate on one of our HPC installations is it would run as a Slurm job, Slurm being the scheduler that decides who gets access to what computers at what time. So it would run as a Slurm job, reserving a certain number of nodes, and then the WDL runner would happen inside that environment. I'm like I said, I'm not positive. I don't think we're running any right now, but that's certainly something that would be, the system would be amenable to operating.
Okay. Jay, this one's for you. Will TAhoma have a routine role in EMSL's automated sample analysis systems that are under development?
I certainly believe that it will. There's a lot of data processing that will happen in a variety of automation contexts. The role that it plays may evolve over time as things like edge computing get further embedded, as custom accelerators come to the fore for a variety of deep learning or AI, other AI, sort of related tasks. But certainly in early days, I would expect that we could play a significant role supporting high performance data analysis.
Okay. Jay, for the large scale research funding for 24 months, is it important to focus on biogeochemical processes in the environment? My expertise is only in geochemical processes in the environment.
I would need to know a little bit more about the details in order to be able to talk about scope. The scope of the large scale research interests change from year to year. So, I can't give a firm answer on, an arbitrary day.
Okay, I think this one's for Kurt. How does TAhoma compare with Qiagen's CLC genomic workbench, where there are already known data sets? Does TAhoma have any advantage over any similar systems?
CLC Genomics Workbench is a software product. So that would be something, I think it's usually targeted toward like high performance visualization workstations. I'm not sure what integrations that particular company offers, if their software is capable of making use of an HPC resource. But what it boils down to is, at the end of the day, despite being a cluster and carefully designed for parallel workloads, the compute nodes themselves run CentOS Linux, which is a derivative of Red Hat Enterprise Linux. And so software that can be adapted to run to some benefit on Red Hat should be workable on the cluster. Whether that's going to be of any benefit to the user, it really depends on the design of the software.
Okay, and for singularity containers, how do you queue them in an HPC? So I think, Kurt, that might be yours.
Yeah, that you run like any other job. A singularity container can be submitted as part of a regular Slurm job, as you're running the container rather than a specific executable.
Okay. And Jay, what's the scope of the science that would be applicable to the CAM science area, or to the computing, analytics, and modeling science area? That is, what types of proposals are more likely to be accepted?
If we're talking about particular areas of science that are of interest, it is whatever is in the call as written is most of interest. We work very closely with BER program managers to ensure that the user program supports science aligned with our priorities. And if the question is about scope, then that is something that we can talk about in discussions leading into a letter of intent or a full proposal. I appreciate that design of computational experiments is sometimes pretty iterative. It is one of the reasons why we are happy to be able to work with users on and providing staff support, whether that's on the modeling side or on scripting or other kinds of software expertise, so that the project team can focus on the highest impact sort of scientific thinking, and we can work to accelerate that.
Okay. So Kurt or Jay, are you ever planning on adding more nodes to TAhoma?
I defer to Kurt on this one.
Well, it's going to depend on the workload we see and the demand from the user community. Obviously, the more heavily used the resource is, the more demand there is for it. That's going to factor into EMSL's future plans for its compute resources. As to whether we expand by adding nodes to TAhoma, or we take that feedback and experience and fold that into the design of a follow-on system, that's in the future. But for now, we're focusing on supporting the user mission that we've got.
Okay, and I think this is another one for you, Kurt. What's the best way to transfer data from our home institutions to TAhoma?
Globus. When you get access to TAhoma as part of a project, you'll be provided with a link to the MSc user guide, which contains information like that and contact information to make sure that you've got access to experts to help make sure that your data transfer is as smooth and high throughput as possible. But primarily, we like to use Globus for that. It's worked well for us.
Okay, another for Amy. What kind of improvements or adjustments are being made to Icicle, MAME, Demos right now?
A great question. We are in the process of developing an Icicle 2.0 right now, in which in our previous original Icicle, basically what we would have is a permanent file that would be handed off between all of the steps, which I think I saw a question asking about pass-offs on here, where like Icicle would basically just track where all the files are, where all of the files are, and call upon the necessary files when needed at every step. What we're doing with Icicle 2.0 right now is, rather than having an actual file on hand, we're going to be saving these as Python objects so that they're more easily callable and don't take as much space up. On top of that, for Icicle 2.0, what we're doing is making all of these functions much more modular, because we have all these different pieces that could be used very easily. But in the original Icicle 1.0, you kind of just had to follow the workflow or already have a lot of knowledge to dig deep into Icicle to get these tools. And we want to make all of these tools available much more easily accessible. And so that's what we're doing with Icicle 2.0, where basically you can just call upon all of it in a Python script. For main, what we're doing is reconfiguring our scoring method, the different library our different library options and our optimization methods. And I believe we have updates on Deimos, but I don't have them right now. Yeah.
All right. Kurt, does Globus have APIs to automate data transfer if needed?
I'll say that well, Globus does have APIs, but there are, I think the thrust of the question is, can we automate data transfer if needed? And the answer to that is yes. We should be able to do that. We've arranged data transfers with other institutions before, and we've got, again, every situation is different. Every institution has different data protection policies, and we certainly don't want to run afoul of any of those directives. So it's something that has to generally be crafted rather than taken for granted, but we have the skills on the team to enable that kind of work.
All right. Well, that was the end of the questions. Although one of our attendees, who happens to be a lead at EMSL, wanted to, or had put a note in the chat that, so note that secure ID is required for Globus, and those aren't issued by default. So it'll have to be requested by you, the user, in advance. So that's just a follow-up to the Globus questions. And with that, I think we're at the end. It's almost 1 o'clock. So thank you, everyone, for joining us. And we hope to see you at September's webinar. Please make sure to complete the event feedback survey that you're going to receive through your email, because we really want to hear your feedback so that we can make these better and better every month. So thank you. Have a great day.